Hello students, in our last class we discussed about so many things about the protein, and the proteins are made up of amino acids. So in between amino acids we will be having what? The bonding will be present. The bond will be present called as peptide bond. At the same time we also discussed about polysaccharides, or we can call it as carbohydrates. So the carbohydrates are also made up of a long chain of monomeric units. So those monomeric units are simply called as monosaccharides. So in between the monosaccharides again there is a bond. That bond is called as glycosidic bond. And one more is lipid. So that lipid is also made up of a long chain of fatty acids or the glycerols, and last one is nucleic acids. And in today's class we are moving on to the nature of bond, or the nature of the bond present in the monosaccharide unit, the macromolecules. See, I mean to say that whatever you take as a complex molecule, say for example when you take protein, when you take polysaccharide or carbohydrate, or you can take lipid, and last one is when you take nucleic acids. As we know that, so these four are simply considered as a macromolecule or macrobiomolecules. So their molecular weight is more than 10,000 Daltons. Their molecular weight is more than 10,000 Daltons, and they are present in the acid insoluble fraction. See, when you take the protein as a complex molecule, very important to understand, when you take protein as a complex molecule, so that complex molecule is made up of very simple monomers, nothing but amino acids. They are made up of amino acids. So that when you take these two words, protein is nothing but complex molecule and amino acids are nothing but very simple. So that the long chain of amino acids nothing but protein, and the protein undergo metabolic reactions resulting in the formation of monomeric units called as amino acids. So simple example to understand nature of bonding. So when you take protein, imagine any kind of protein. Protein nothing but, many times I told you people, the word protein refers to, it is a complex food molecule. That complex food molecule is made up of long chain of amino acids. Long chain of amino acids. So example, some of the protein contain 100 amino acids, some contain 1000 amino acids, some contain several thousand amino acids. That is different. I mean to say, the number of amino acid is going to differ from protein to protein. So in our nature there are several thousand different types of proteins are there, and their monomeric units or the number of amino acid will be definitely different. So in my example, this is considered as a protein. That protein is made up of several amino acids. So each consider as amino acid. Imagine, each consider as amino acids. The thing to understand is, I mean to say, what I am trying to tell you is that these two amino acids are held together by which bonding? How they are held together? Yes or no? So that is my question here. And I can tell you people, so in detail, imagine this is what? Yes, this is typical amino acid. This is one typical amino acid. Same time, when we can understand again, this is another amino acid. Yes. So now when you take these two, so called as dipeptide. Dipeptide means there are two amino acids are there. Now, so what we are discussing here is that these two amino acids are, I mean, how they are held together. This is the main question here. How they are held together. I mean to say, which bond it is. Which bond it is, I mean, so which bond is present in between two amino acids. That is we are going to discuss here. So here NH2 means how can we write this? Yes, this is how. So nothing but NH2 or NH2N. Here what is going to happen? In between there is a bond. Yes, so that bond is simply considered as peptide bond. Please do remember, one last question, which bonding is present in between two amino acids? That is peptide bond. So that peptide bond is formed by the elimination of water molecule. Elimination of water molecule. Here see H2 and here from O. So nothing but elimination of water molecule. So that meioty is considered as dehydration. Remember the word, we can call it as dehydration. So again, D refers to removal and hydration refers to water. I mean to say that this peptide bond is formed between two amino acids by elimination of water. So only when the water is going to eliminate, the bond can be formed. So this is nothing but the peptide bond. And most very important thing to understand is this peptide bond is formed between these two amino acids. How? That is, the carboxyl group of one amino acid, remember, so the peptide bond is formed between two amino acid. How? That is, carboxyl group of first amino acid and amine group of second amino acid. This is how we need to remember. See how the bonding is formed here? That is, the carboxyl group of one amino acid with the amino group of second amino acid. So this is what the bond formation in case of, that is protein. That is called as, see the first answer. First answer is peptide bond. Over. So what are the things we need to understand? The protein, nothing but it is a complex food molecule which is made up of long chain of amino acids. In that, two amino acids are held together by which bond? That is, by this presentation we can understand this is one amino acid, second amino acid. See, the carboxyl group of one amino acid or first amino acid can form the bond with the amine group of second amino acid. Yes, that too elimination of water molecule. The process is called as dehydration. So that now the bond is called as peptide bond. This is about protein. Very simple. Next. Second one, when you take polysaccharide, is polysaccharide nothing but carbohydrate. Now tell me, what is polysaccharide or carbohydrate? Again, like protein, it is a complex food molecule and it is made up of monomeric units. Monomeric units. So those monomeric units are nothing but we can call it as monosaccharides. We can call those as monosaccharides. So the word monosaccharide refers to, I told you yesterday, I mean in the last class, mono refers to one, saccharide refers to sugar units. Sugar unit. So monosaccharide refers to only one sugar unit. Monosaccharides refers to many sugar units. Yes or no? So here polysaccharide refers to many monosaccharide units. Many monosaccharide units together, it can make polysaccharide. So in the polysaccharide, what kind of bond is present? Imagine, I can again explain you people, polysaccharide. Imagine in the polysaccharide, I will take small example, that is we'll take disaccharide. Yes, so here monosaccharide refers to only one sugar unit is there. In case of this one, there are two sugar units are there, hence called as disaccharide. See, there are two sugar units. Yes, it is 1 sugar unit, and again here we will be having second sugar unit. In this, first carbon atom, second, third, 4, 5, 6 or whatever, and here also it is 1, 2, 3, 4, 5 and 6 somewhere. Yes, imagine these two are nothing but sugar units. I mean to say that there are 6 carbon atoms, means simply called as hexose sugar. These two are simply called as hexose because there are 6 carbon atoms are there. So the structure which contains 6 carbon called as hexose sugar. See, in this the bond is formed by, I mean to say that there are 2 sugar units are there. Nothing but disaccharide. Very simple to understand. So in disaccharide, how these 2 monosaccharides are held together, that is our question. Yes or no? So this is what our question. In this, here we will be having OH, again he will be having OH. In that, this bond is formed by again removal of water molecule, that is OH and H, nothing but H2O. So when you remove H2O, that is water from this, so the bond will be like this. Yes or no? So now this is called as remember, glycosidic bond. This bond is called as, I mean to say, the bond which is present in between 2 glucose units or 2 monosaccharide units is called as glycosidic bond. Very clear to understand? And next, so which kind of glycosidic bond? I mean, where it is going to form? That is, the glycosidic bond is formed between 2 monosaccharide units, that too, first carbon of first glucose and fourth carbon of second glucose or second monosaccharide. First carbon of first monosaccharide, fourth carbon of second monosaccharide. Hence it is called as alpha 1,4 glycosidic bond. 1,4 glycosidic bond. Clear my point? Alpha 1,4 glycosidic bond. Why it is 1? The first carbon of first monosaccharide, fourth carbon of second, that is monosaccharide unit. Hence it is called as alpha 1,4 glycosidic bond. So my final conclusion is that in case of polysaccharide or carbohydrate, the monomeric units or monosaccharides are held together by alpha 1,4 glycosidic bonds. Very easy to understand. See this structure. This is second one. Yes, final answer is alpha 1,4 glycosidic bond. First one is peptide bond, second one is alpha 1,4 glycosidic bond. Simple. Next, in case of lipid, again in the lipids you will be having definitely, simple answer is ester bond will be present. Remember, in case of lipid, when you take arachidonic acid, yes or no, palmitic acid, lecithin, whatever the example you take, so there definitely the ester bond will be present. And one more, that is nucleic acid. Sorry, in case of lipid we will be having fatty acids. So these fatty acids are held together by ester bonding or ester bond. Next, last one is nucleic acids. Nucleic acids or nucleic acid. I told you in the last class that is DNA and RNA together called as nucleic acids. Here nucleic acids refers to remember the word polynucleotides. The word of polynucleotides refers to, again, poly refers to many, nucleotides refers to, I told you in the last class, that is nucleotides. There are many nucleotide units can make nucleic acids. Long chain of nucleotides, nothing but polynucleotides. See, here my question is, I mean to say, what we are discussing here is that here we have one nucleotide. Imagine. Here we have one nucleotide. Same time, here we'll be having one more nucleotide. So the question is that these two nucleotides are held together by which bond? So which bond is present in the two nucleotides? We'll discuss here one by one. Here, first one is, we discussed in detail in the last class. So first carbon atom, second, third, fourth, CH2OH, that is fifth carbon atom. Yes or no? This is what our simply called as pentose sugar. Simply called as pentose sugar. So in the pentose sugar structure, at first carbon atom, very important, at first carbon atom, here we will be having nitrogenous base. Nitrogenous base. Now the formula is pentose sugar plus nitrogenous base, which yield nucleoside. And at third carbon atom, I mean to say at this position, we will be having phosphoric acid in the form of phosphate. Yes, pentose sugar plus nitrogenous base, which gives nucleoside plus HPO4, which gives nucleotide. Can you understand the structure? And here, yes, again we will be having one more pentose sugar. This one must, I mean second pentose sugar, you will be having CH2OH at fifth carbon atom, and here 1, 2, 3, that is at third carbon atom, phosphate, and first carbon atom you will be having again one more nitrogenous base. See, how to remember and what to remember is that in this structure, the pentose sugar binds with nitrogenous base, that is the pentose sugar nitrogenous base. These two are held together by glycosidic bond. Remember, yes, glycosidic bond, that is the bond present in between the pentose sugar and nitrogenous base is called as glycosidic bond. Same time, I mean to say, I am telling you people this one, which bond it is? Glycosidic bond. I have mentioned there. The bond present in between pentose sugar and nitrogenous base is called as glycosidic bond. Next question is, here at third carbon atom, we will be having phosphoric acid. Yes, phosphoric acid. So this phosphoric acid is formed in between, that is, I mean to say, this structure is called as nucleoside. Yes or no? So that nucleoside combines with the phosphoric acid by means of which bond? Ester bond. Yes, ester bond, that is the bond present between the pentose sugar and nitrogenous base is called as glycosidic bond, and the bond present in between the pentose, that is nucleoside, and phosphoric acid or phosphate is called as ester bond. Finally it yields nucleoside. So together called as one nucleotide. And again here we will be having one more nucleotide. Yes or no? One more nucleotide. See, now my question is that polynucleotide means long chain of nucleotides. See, one nucleotide held together by another nucleotide, that is, this is formed by third carbon of one nucleotide, and fifth carbon of second nucleotide. This is one and it is second. How the bond is going to form? Third carbon of first nucleotide, fifth carbon of second nucleotide, like this. Clear? And this is called as which bond? Phosphodiester bond. See, in the single structure, we'll be having 3 bonds. One is, see, how to remember? Pentose sugar plus nitrogenous base. Which bond? Glycosidic bond. This is first one. Same structure. Second one, see here, this is over. Next one is nucleoside and phosphate. Next is nucleoside plus phosphate, that is HPO4. There the bond is, which bond it is? Ester bond. Next one is ester bond. And last, very important, is nucleotide plus nucleotide. Nucleotide plus nucleotide. I mean to say, one nucleotide hold second nucleotide by means of which one? Phosphodiester bond. Phosphodiester bonds. So this is our today's topic. I hope you understand the whole concept. So in protein, peptide bond, and polysaccharide, that is glycosidic alpha 1 for glycosidic bond, then lipid, ester bond, then nucleic acids, that is there are 3 bonds. One will be glycosidic bond, ester bond, and last one is phosphodiester bonds. So these all different bonds together, they can make the complex macromolecules like a protein, polysaccharide, then lipid, and last one is nucleic acids. I hope you understand this concept. Next, yes, so we'll continue the same topic, that is nucleic acid. Till now we discussed, so I can recap very quickly. Nucleic acid, that is either we can call it as DNA or RNA. Yes, deoxyribonucleic acid and ribonucleic acid. So these two together called as nucleic acids, and they are present within the cell, that is nucleus. In this, just now we discussed about that bond is, which bond is present in the nucleic acids. So during formation of that, how the nucleic acid is formed, we'll take DNA. How the DNA is formed, that is first one is structure. How the DNA structure is formed, that is best is secondary structure. DNA is secondary structure of DNA is present. So how the DNA structure is present, we'll discuss some important topics. That is, first one, we need to understand what are the different chemicals present in the DNA. What are the different chemicals present in the DNA? One is, I told you, that is pentose sugar, yes or no, also called as, simply called as ribose sugar. Simply called as ribose sugar. Second one is nitrogenous bases. So those nitrogenous bases are and those are purines, pyrimidines. Purines are of two types: adenine, guanine, cytosine, and thymine. I told you. See, and third one is HPO4, that is phosphate. Actually, that is phosphoric acid. It is in the form of phosphate. So these three important chemicals are always present in the DNA. Yes or no? So, and this is enough for today's class. And how the DNA is present, how it is formed, what are different chemicals are arranged, how they are arranged. So that thing we'll discuss in tomorrow's class. So thank you very much.